Hello everyone. Today for the Polar lecture series, we invite the Professor Sophie from University of Orléans or
Orléans.
Orléans. Orléans. Okay, and the title is Multiphase Flow and Reactive Transport in Porous Media, an experiment microfluidic approach. Thank you. And Sophie, now it's your turn.
Okay, so thank you for Thank you for the introduction and thank you for the invitation. So yes, today I will show my recent works about multiphase flow and reactive transport in porous media. Oops, is it working? Okay, oops. So before I started, I will also take this opportunity to introduce myself and to introduce my my background. So I did a PhD in fluid dynamics, where I was studying red blood cells flow in microchannels. So this is like similar to problematics of colloid transport in porous media. And then I did a short postdoc in France, where I was studying transport properties of carbon nanotubes using a microfluidic device. And then I spent 4 years at Stanford University in the Department of Energy Resources Engineering in the group of Tony Kovsketch, where I applied all my skills in microfluidics and dynamics of flow to geological porous media, where I was studying multiphase and reactive flows using micromodels. And then I joined the University of Orléans in 2017, where I'm still working on multiphase and reactive transport in geological porous media. So our lab ISTO it's an Institute of Earth Science of Orléans. So Orléans is located 1 hour south from Paris in France. So it's a nice city. And within this lab, I'm part of the Porous Media Group. So in our group, we are interested in multiphase and reactive transport processes in porous media that are linked to, for example, to the process of CO2 injection and sequestration in geological formation. So for example, the process of CO2 storage involves CO2 that is injected in a porous reservoir at sufficiently high pressure and temperature, so it's in a supercritical state. And the CO2 will displace native fluids, so usually brine, from the pore space. So this drainage process results in some CO2 that will become trapped as droplets or as ganglia in the porous matrix. And also the trapped CO2 can dissolve through the brine and results in an acidified aqueous phase that will transport, for example, acid to the mineral surfaces, and with following mineral and dissolution precipitation. So also challenges associated with CO2 sequestration are to be able to assess the storage capacity of a reservoir, to optimize the injection process, and of course to ensure safe and sustainable storage underground. Other processes that are that also involve different fluids and multi-feed flow problem are contaminant transport in aquifers, for example, where you also have immiscible phase flowing through a porous media. And here one of the challenges is to find a way to clean up contaminated aquifers. So for these processes, some challenges that we try to address and that are linked with these processes are the following. So we want to be able to capture the complex space and time evolution of the interfaces, meaning when we have breakup of interface or matching of interfaces. And this will help us to predict the amounts and location of residual trapping in a porous medium. As I will discuss later in this talk, we want to see the effect of momentum transfer between immiscible fluids in porous media, and I will try to show you why it's important. Also to assess the role of pore-scale events on macroscopic energy dissipation during two-phase flow. And regarding reactive transport, one of the main challenges is to evaluate the consequences of mineral precipitation or dissolution on the evolution of the macroscopic transport properties, like how porosity or permeability evolve. So to address this challenge, we adopt a multi-scale strategy because ultimately we want to understand and predict processes at the feed scale, but these processes depending on what is happening at smaller scale, like the core scale, and this is linked with pore-scale events and also molecular, what is happening at the molecular scale. So I am particularly interested at looking at the mechanisms at the pore-scale and to understand their consequences on macroscale behavior. So to study these mechanisms, in our group, we combine numerical simulation, experimental modeling, and also characterization and acquisition. So regarding experimental modeling, to get insight on the processes happening at the pore scale, we perform micromodels experiments. So basically, micromodels are a 2D representation of porous media that allow to have a direct visualization of the pore scale mechanisms. So classically, a pattern is etched on a substrate, and the substrate is covered by a glass plate. The material that we use are usually silicon and glass or PDMS. So PDMS is silicon elastomer. And the patterns that we etch can be of very variable geometry and very diverse also complexity, from very simple geometry made of single pores or successive pores, constriction, pore doublets or dead-end pores, to geometry where we have cylindrical pillars that are distributed in a media, and to geometry more representative of real work patterns. So here we have geometries that are drawn from samples of here sandstones and here from a carbonate. So you can etch this geometry on your micromodel. When I was at Stanford, we also developed a technique to be able to have dual depth micromodels, where we had a smaller depth for the, so it was for the carbonate, kind of very heterogeneous porous media. So we had smaller depths for the micropores and larger depths for the macropores. So kind of 2.5D micromodel. And we can even include real rocks into the micromodels, and I will talk about that later in this talk. So with these micromodels, this is an example of one of our experimental setup, but it's more or less the same for all the experiments. So the micromodels are placed on a holder to deliver fluids. The micromodels is placed under a microscope and linked with a camera for flow visualization. And the fluid, so we have a syringe pump to inject the fluids at the desired flow rate, and the fluids that we use can vary from aqueous phases to oil, different oil, to gas, or even reactive fluids for reactive transport. So with this kind of setup, we record sequences of image of the flow. And one of the, so with this setup was one of our first question was to ask us how quantitative are we when we perform these experiments. So to be able to measure the velocity fields during micromodels experiments, we optimized and we validated a technique that is called micro-PIV to be used for complex porous media geometry, like here, the sandstone micromodels. So the principle of this technique is to put microparticles, so here they are 1 micron in diameter, to put them in here in the water, and so the particles will follow so they are chosen so that they will follow the flow without disturbing it. And so this is an example of image sequence where you can see the particle flowing around the grain. So this is for a single phase flow in a sandstone micromodel. And with this sequence of image, we can use this sequence of image to perform micro PIV measurements and to obtain velocity fields during an experiment. So to validate our measurements, we compared our PIV results with numerical simulation for single phase flow. Because for single phase flow, the models now are well understood, and they serve as a tool to validate our experimental measurements to be sure that we were measuring the actual velocity of the flow. So here you have just one example of our results where we compared micro PIV measurements with numerical simulation. So this is for a small area in our micromodels, about 50 micron width. So we have so here this is a magnitude of the velocity, the displacement along x, and the displacement along y. So we have a pretty good agreement between experiments and simulation. Here you can see the velocity profiles plotted along the white line. And here you can see examples of velocity vectors. So basically we are able to obtain a resolution of velocity vectors of 2 microns, meaning that we have one velocity data every 2 micron in the micromodels. So with this technique, so we really are going towards a better understanding of flow mechanisms at the pore scale, especially now for cases where numerical models are still in development. And especially these techniques allow for a quantitative comparison between experimental and numerical data. So now with this setup, I will present two main projects, I will say. So I will start to focus on two-phase immiscible flow at the pore scale, and then I will show some of our new results on reactive transport, so coupled hydrogeochemical processes in porous media. So for two-phase immiscible flows, so here I write the equation of two-phase immiscible flow at the continuum scale, so at the Darcy scale, where each fluid phase here is treated separately with its own equation for describing the balance of mass and momentum. So here you have the volume fraction of, so for if it's oil and water, for example, you have the volume fraction of oil and water, the pressure of oil and water, and you have the drag force that is exerted by oil or water on the solid surface, and you have the mutual momentum transfer between the weighting and the non-weighting phase between the oil and the water. So this formulation is generic, and you have more details in our recent paper here, and this equation can be rewritten in terms of the standard generalized Darcy's law with relative permeabilities. So here this is so the standard model that is used in most of the engineering process using two-phase flow. So here in this formulation, the fluid immobile is assumed, the fluid interface, sorry, is assumed to be immobile, meaning that here in this formulation, these terms, the mutual momentum transfer between the two phases are assumed to be negligible. However, what we have observed is that maybe these terms are not negligible in some cases. So why we say that? Because with some experiments that we did, so this one is an experiment in sandstone micromodels for two-phase flow. So we did a drainage. So it was a drainage for with oil and water, and with condition close to a system of brine and CO2 in geological formation in terms of viscosity ratio and capillary number. So for this experiment, what we observed is that during the drainage process, we have some, so here we had a globule of water, so you can see here. We have water that is trapped between the solid grains and between oil, and the oil is still flowing. And this water, so thanks to the micro PIV measurements and thanks especially to the particles that are seeded in the fluids, we can see that this water is recirculating. So usually you would assume that a fluid that is trapped during two-phase flow is immobile, but what we saw is that it's still flowing, and it shows recirculating motion. So here I plotted the streamlines thanks to the PIV setup. So this is a driven cavity flow that is due to the shear stress at the water-oil interface, and that is due to the oil that is still flowing. So here you can imagine that the oil is still flowing. So now the question is, okay, is this mutual momentum transfer really negligible? So we can imagine that if we have, for example, lots of trapped globules of water that are recirculating, maybe these viscous coupling effects are not negligible. However, it's not clear what is the exact meaning of these coupling terms. And for example, we don't know if they really depends on the viscosity ratio or not. And the importance of these terms has been questioned for more than 60 years now. So one of our objectives was to use, to realize some macromodels experiments, so some flow measurements directly at the pore-scale, and we wanted to try to evaluate the importance of this interfacial momentum transfer at the fluid-fluid interface. So for that, we did some drainage experiments. So we used very simple geometry. So the idea was to try to really isolate the flow mechanisms and to control all the flow parameters. So we had various geometry with single pores, with dead-end pores or pore doublets, and we did some drainage experiments. So the microchannels were first filled with water, and then we are flowing oil at different flow rate for different viscosity ratio. The water is seeded with microparticles to do micro PIV measurements. We measured the velocity at the oil-water interface during the invasion. And with this velocity, so we can measure so we have two main macroscale parameters that we are using and that are characterizing the flow. The capillary number that measures the ratio between viscous forces and surface tension forces, and the viscosity ratio, so the ratio between the viscosity of the oil and of the water. So with this setup, when we have a globule or droplet of water that is immobile in the media, we measure, like here, for example, we measure the volume of the trapped water. We measure the fluid-fluid interfacial area. We can measure the velocity field in the, so if the water is moving in this immobile globule, we can measure the velocity field. And we also measured the local rate of dissipation of mechanical energy that is depending on the velocity. So why we measure that? It's because the rate of, so the viscous coupling, and the local rate of dissipation of energy is associated with the mobility of the phases and with the overall dissipation of mechanical energy in the system. And we also measure the shear stress at the interface. So we did that for a lot of different experimental conditions, and I will show now some of the results. So to show you some example, here you have two videos for a drainage in this kind of setup. So this is for two different viscosity ratio and two different capillary number. And this is for a geometry representative of dead-end pores. So here you have the water seeded with microparticles, and we inject oil. So here in this case, the water that is trapped is immobile. And here for another experimental condition, we also have water and we flow oil. And here you can see we have recirculating motion of the water that is trapped in the Dandan pore. So for this case, we can measure the velocity field with micro PIV. So here, we have this driven cavity flow that we observed. And so by doing this kind of analysis for various flow conditions and geometry, what we show is that we were able to identify 4 different regimes of viscous dissipation. We had identified them based on the profile of the velocity profile and the profile of dissipation of energy inside the trapped phase. So for example, for each cases, for each regime, we plot along the yellow line that is perpendicular to the interface, we plot the velocity profile, and here this is the local rate of dissipation of energy in the trapped phase, and this is the same for the other regime. So first we have a first regime where the trapped phase is immobile, so we don't have any momentum transfer at the fluid feed interface. And then we have 3 other different regimes. So for example, for this regime that we call rim globule interface driven, so this is when a small globule of water is trapped. And for this regime, the center of mass of the recirculation is the same as the center of mass of the globule. And in this case, the dissipation is maximum at the center of the trapped phase. And we have another regime that we call lead cavity driven. That is when we have large pockets of water that are trapped. And in this case, most of the velocity, so most of the recirculation and most of the dissipation is located at the vicinity of the interface between the fluids. So what are the implications of these different regimes at larger scale? So what we identified is that the trapped phase is immobile when the non-wetting phase, the oil that is flowing, is more viscous than the water. So this is in agreement with previous studies. And for the other cases, when we have a viscosity ratio close to 1 between oil and water, we saw that, and especially for low capillary number, we show that in lots of cases, the momentum transfer as a fluid feed interface is not negligible. And what we also have shown is these different regimes will depend on macroscale parameters. So they will depend on the capillary number, on the viscosity ratio, and also on the geometry of the pore, and especially on the interfacial area and on the geometry of the trapped phase. And they will also depend on lubricating effect, and I will just show you why here. So in particular, we have shown that this effect is linked with lubricating films. So when you have a drainage in a water weight micromodel, during the oil invasion, you always have some films of water that are remaining on the pore, on the wall of the pore. And here we have 2, so the importance of these films is shown here, where we have 2 experiments where it's for the same viscosity ratio, close to 1 between oil and water, and for a different capillary number. So for the higher capillary number, we have a trap phase that is immobile. And for a smaller capillary number, we have a recirculation. And what we clearly see, so maybe you can see it here, is I can clearly see the importance of lubricating films and corner flows in this case, because we see some particles that are actually flowing from the corner flow to and then linked with the trap pocket here and linked with the recirculating motion. So what we think is that these corner, so these lubricating films, they are connecting the trap pocket to the main pressure in the pressure in the channel. But for us, what is still not clear for now is why, in this case, for a lower capillary number, we can clearly see these films and this effect, and why we don't see it for a higher capillary number. So these are some results that we are still not fully understanding them. So now I move on to another two-phase flow phenomena for which also lubricating films are important. So when
Sophie,
yes.
Before you leave the topic you just talked about, my name is Bjorn, by the way. Can you go back one slide, please?
Yes.
The number 17. In the figure to the left, where you have trapped water, what is the flow pattern in the oil phase there? With the low capillary number, you could easily see that the oil is flowing also in the sort of stagnant part of the down into the pore there. But with a high capillary number, I don't see the oil phase.
So yeah, here we cannot really see the oil flow because we don't have any particles in the oil, but clearly here at the interface, so this is not a movie, but here at the interface we can clearly see that the velocity is 0. So we think that it's for sure it's moving here, but then it looks like the water is completely disconnected. And here the velocity at the interface is just 0. So it's just acting as a solid wall here.
Right. So it's not only a question of the coupling parameter, it's also a question of the flow pattern in the flowing phase locally near the interface. Yeah.
Yeah, of course.
Okay.
Yeah. That's why it's very complicated.
Thank you.
And I also have a question regarding to this experiment. Have you run any other experiment similar to this with different width of the like the cavity or not? But it might also affect that film flow.
So in this case, the cavity. No, the shape of the cavity was always the same, but then we have other geometry. I did not present all of them, but we have like construction where we have trapping or pore doublets, so first thing we can see that it's difficult to replicate exactly how it will get trapped and the shape of the trapped phase. But yeah, it will be interesting to see the effects of having different depths of the cavity or different geometry.
And what about in a three-phase case? I mean, for example, you have one phase at the bottom of the cavity, another one at the middle, and another one is flowing on top of that. Don't you think that this interaction, I mean, this momentum interaction between different phases are affecting that film flow?
Yeah, I think in some cases it can. Yeah, if you have transfer between all the interface, yeah, it can play a role. I guess so.
A question from Carl Fredrik Berg here. You had an earlier slide where you showed this water stuck in the corner before a constriction, and you also had recircling flow there.
Oh, yes.
Yes. Do you see films in this case too? Or?
Yes. Yeah.
Okay. So whenever you see this recirculating flow, you see that there are films.
Yes, exactly. Yeah. And as we've seen, and in the first experiment in the sandstone micromodels where we first observed this phenomenon, we can also see the films, and we can also see particles going through the films. So yes, when we have recirculation, we have the films.
Yeah, so you have no examples of sort of momentum transfer over through the interface which are driving this circulating flow?
Sorry?
Because when you have those films, the circulation of this flow here might be because of the water coming in and driving the flow, right? It's not necessarily a momentum transfer over the fluid.
But we can, yes, but then but we can also clearly see at the interface. We can also clearly see the velocity at the interface and to drive the flow. Yeah, in such, like for example here, deep cavity. Here we can clearly see that it's linked with really the interface also. But I agree that it's probably a combination of both.
Okay, thank you. You have a very nice picture, by the way. It was
Thank you.
very nice to see. Thanks.
One quick question. Is the water as wetting phase or non-wetting phase?
It's a wetting phase.
So oil is non-wetting phase, right?
And so here, yeah, water is and it will be actually really interesting to do same kind of experiments where we trap the non-wetting phase to see if we. But for now we are not really able to see the non-wetting phase with microparticles. But it will be interesting to see if we still have raw circulation in this case.
And normally the PDMS material is a hydrophobic surface.
Yes, so what we do, so we use a plasma treatment to make the surface hydrophilic.
Okay.
So before your experiments, you can treat it with plasma and it stays for a couple of hours or days. So yeah, so actually, oops. So this is still linked with films when you have the breakup of bubbles in pore throat. So this is referred as the mechanisms of snap-off. So here in these mechanisms, the idea is that the non-wetting phase, so gas, or it can also be oil, becomes unstable at the constriction, and at some point you can have a bubble that will snap. This process of snap-off has some implication for CO2 storage because it's assumed to be one of the main trapping mechanisms, but some questions are still remaining regarding these phenomena. So for example, we don't really know what is the dynamics of the liquid film during the motion of the bubbles or of drops. We don't know exactly the dynamics of the length here of water that just after a snap-off, for example. And also there is some question about roof criteria. So in most numerical models, this is a static criteria that is used to predict snap-off that is based on mostly on the geometry of the pore space. But some questions are remaining on this criteria, and especially to include dynamics effect like the capillary number. So what we did to study this effect was to perform some experiments. So now the experiments are in a circular capillary tube with a constriction. So the tube, and for these devices, we performed some drainage experiments. So the tube is initially filled with water in blue, and then we injected air. And for this experiment, we compared the experiments with numerical simulations that were developed by Mortaz Abou Al-Saoud at Stanford during his PhD. So, and we have a blue dye here in the water because we wanted to, initially we really wanted to get insight into the thickness, so the dynamics of the films on the wall and the thickness of the films. So here you can see that you have some blue dye, and we were actually able to link the intensity of the dye to the film thickness, and then to perform some dynamics measurements of film thickness. So here you have an example for a capillary number of 2 to the minus 3 of simulation here and experiment here. So you can see the film that is growing at the construction, and at some point that will snap a bubble of water. So especially what was really interesting for us is that we observed a phenomenon of repeated snap-off and reconnection so we saw this phenomenon both numerically and experimentally. So here you have and we observe that for different capillary numbers. So here you have the measurements of the film thickness in the construction for experiment and for simulation. So here when we are at 1, it means that we are filled with water, and when it's minimum, it's filled with gas. And we can see this repeated here snap off and reconnection and snap off and reconnection. So both experimentally and numerically. So first, based on these experiments, we were able to find a correlation between the time to break up a bubble, meaning the time between the invasion and the first snap off. So there was a correlation between this time and the capillary number that is in agreement with previous theoretical studies. And we were also able to, so for these cycles of snap-off and coalescence, we were also able to show that the period of this snap-off coalescence is also depending on the capillary number. So this phenomenon might have implication for residual trapping. For us, it's very interesting, and especially because now there have been recent studies that observe that, so in more complex porous geometries, observe that during two-phase flow, especially at low capillary number, you can have pathway of the non-wetting phase that will disconnect and reconnect, and this has been called intermittent pathway flows. And these are phenomena that still need a deeper understanding of especially of the dynamics behind intermittent pathway. And also we still need to understand the consequences of these phenomena or intermittent pathway on large scale properties and how to get a better prediction, for example, of residual saturation. So this is still something that we are interested in, and we still have some questions. What is also interesting for us is that we observe the same cycles of snap-off and coalescence in micromodels of rectangular cross-section. So here you can see a snap-off, and then as we keep injecting, we have a coalescence and snap-off again, and coalescence again. So we still need more experiment to characterize this phenomena, especially to identify maybe different regime. But it is really interesting. So to conclude on this first part, what two-phase immiscible flow in porous media, what we have learned from that is that first at the pore scale, so as just said, we identified cycles of snap-off and coalescence in constricted capillaries. We also identified that during drainage, the trapped wetting phase can show this recirculating motion, meaning that we have a slip at the fluid-fluid interface and that we have viscous dissipation. And we identified different regimes of viscous dissipation. So what are the consequences of that at larger scale? So for snap-off, the consequences are probably on the residual trapping and on the modeling of two-phase flow. For the viscous dissipation has consequences on the mobility of the phase and of the flowing phase in our case. So we have identified that in some cases, the Darcy extended law for two-phase flow can neglect the momentum transfer, and in some cases it probably cannot neglect it. And for future work, we still need to have further insight into trapping mechanisms, still need to have an accurate knowledge of lubricating films. And we also want to investigate the consequences of this fluid-fluid interaction, for example, on mass transfer. Because you can imagine that this recirculation will enhance the mixing during the recirculation and have some implication. So, so now I move to our study on coupled hydrogeochemical processes.
Sophie,
Yes.
this is Björn again. Can I ask you another question?
Yeah, sure.
By the way, this is a very nice and very interesting talk. Can you go back one slide, please?
Yes.
About the snap-off. You have visualized the snap-offs. Can you also hear them? If you had a very sensitive microphone, could you listen to a sound and correlate that to the visual recordings that you have?
Good question. Yeah, I don't know. What do you mean?
Because
yeah.
if you could,
yeah.
you could, if you find the correlation between the two, you could also do such measurements in systems that you could
more
not
complex
do.
systems. Yeah. Yeah, it will. Yeah, it will be actually. It will be nice, and yeah, I guess can be correlated to. So, for example, or maybe it's just correlated with the pressure signal, but it's true that in microfluidics experiments, we cannot really get the pressure between even inlet and outlet of our micromodel very accurately, but that's a nice idea to try. So for reactive transport, one of the main objectives is, as I said before, is to evaluate the consequences of mineral precipitation or dissolution on the evolution of the porosity and on the permeability of the porous media. And this is linked with the evaluation of the accessible surface to reacting species. So for example, here, you have a Darcy scale representation, and for each voxels, you have average properties over representative elementary volume, like permeability or reactive surface area. Now, if you zoom in and that you look at the velocity fields at the pore-scale, you can see that the flow field is heterogeneous with preferential flow paths. So these are some simulations. And you have preferential flow paths that developed, and you can imagine that if you inject an acid in this domain, you will not reach all the grains equally. And this has to be described as a macroscale parameter. So, and this is some of the questions that we have for reactive transport. So when I started to focus on reactive transport, we actually started with a setup where we investigated the dissolution of a single grain of calcite that was in microchannels, and this way we were able to look at the effect of flow rate and of the activity of the fluid on the dissolution of the shape of the calcite during dissolution. So these were very simple experiments, but that can bring very high accuracy measurements, and that we used to validate the models. Today I will not present that, but however I will show you one of our very recent work where we developed new kind of micromodels and new kind of measurements to look at this coupled reactivity and transport phenomena. So this has been published recently in Lab on a Chip. So this is a work that we did in collaboration with Jenna Bonosami, that is a researcher at the Jülich Research Center in Germany. So this is a setup. So we have micromodels, and these micromodels consist in a reservoir here, and we fill this reservoir with reactive grains. So in our case, the reactive grains are celestine, strontium sulfate. So we fill them with grains of desired size and desired mineralogy. So the grains are injected through this crystal injection channel here, and once we have injected the grains in the reservoir here, we close this channel, Then we have inlets and outlet channels, and we have an upstream and downstream porous media to have a uniform flow through the reservoir filled with crystals. So the reservoir is about 100 microns per 800, something like that. So here you can see, so this is a chip that is made of PDMS and glass. And here you can see a picture of the chip where here in white, these are the grains, the celestine grains in the reservoir. So with this setup, so first we inject the grains, then the injection channel is closed, and then from the inlet to the outlet, we inject, in our case, a solution that is saturated with strontium sulfate for 1 day, so that we are sure that all the grains are stabilized in the reservoir. So here, these are some pictures, so optical picture of the medium that we can get. So with this process, we can create homogeneous or heterogeneous porous medium. So here you have a picture of the homogeneous porous medium. So this is a wall reservoir, and in white, these are the reactive grains. And here this is a zoom at the inlet. And we are also able to create a medium with the presence of a fracture. So here you have a fracture, and here you have a zoom in the fractures. So we investigated these two kinds of porous media for the same reacting system and for the same injection rate to see the effect of the heterogeneity of the system on the reactivity. So here, this is the experimental setup. So our micromodel is again placed under a microscope. We have a camera for optical visualization, but most importantly, in this case, the microscope is also coupled with a Raman spectrometer. And this Raman spectrometer gives us in situ and in real time monitoring of the details of the chemical composition of the system. So this is quite new information and very interesting setup for this kind of process. So we have at the inlet of the sample so after injecting our grains of strontium sulfate, we started the experiments. For that, we injected a solution of barium chloride at 1 millimolar, and so it's at 500 nanoliter per minute. And we did the injection for 60 hours for both type of medium. Then the reaction that will happen is this one, where your crystals of celestine will dissolve, and we will have the precipitation of barite or strontium barium sulfate barium. Sorry. And we will see how this reaction occur and depend on the flow parameter. So we checked the mineral distribution and some optical picture of our macromodel during the 60, so at different times during the 60 hours of injection of the reactive fluids. So with that, we are able to, I will show you, to get details on the mineral distribution in the system. We also can get a 3D representation of the pore structure, and I will show you how we can see the influence of the hydrodynamic heterogeneity on the reactivity of the minerals. So here are the mineral distribution for the homogeneous porous media. So this is so at T0, this is an optical picture of the media at the inlet. So with some zoom at the inlet and outlet. And here at the bottom, this is some 2D Raman imaging of the homogeneous porous media after 60 hours of reaction. So here you have in blue, this is a PDMS. In red, this is the initial crystals, the celestine, and in green, this is the barite, the crystals that has precipitated. So what we see for this homogeneous porous medium is that at the inlet, so the inlet is here, at the inlet we have about 50 micron wide precipitation front here. And at the upstream of the and at the outlet of the reservoir, most of the celestine has not reacted. And here we have a zoom in at the inlet. You can see, and out the outlet, most of the celestine is still there. Now, if we look at the mineral distribution for the heterogeneous case, now here we have so we have an heterogeneous distribution of the minerals, and after the reaction, what we can see is that we have a higher barite content close to the fractures, and especially here on this right side. If we do so, most of the medium has reacted in this case, and here we have a depth scan, so a scan of the Raman spectrum over the depths of the micromodel, and we can see that we still have some celestine crystals that are surrounded by barite. And we also have so clearly the first observation is that the chemical reaction appeared more pronounced for the heterogeneous media than for the homogeneous one. We can also so here this is for the heterogeneous porous media, where we can see the temporal evolution of the chemical composition. So here at the inlet, so from 0 to 60 hours, and here at the outlet, here at the fracture, from 0 to also to about 60 hours of reaction. What appears here is that the reaction occur mostly in the compacted region and not at the fracture porous media interface. And here we can, for example, follow one celestine crystal in the fracture that will get dissolved during the process and get rounder during the process of dissolution. So with this setup, as I was saying, we can also have stacked images, Raman images that we can collect over the depths of the micromodels to visualize the grains in 3D. So here we visualize it with some tools. This is here for the homogeneous porous media at the inlet, where after the reaction we see that some barite has precipitated. And and here this is the same kind of images with the images at the inlet, but for the heterogeneous porous media. We can clearly see that here all the celestine has been replaced by barite. So the main advantage of these 3D images is that you can measure the porosity and the volume fraction of each mineral that is present. So this is what we show here. So this is for images that are close to the inlet. So for the homogeneous porous medium and for the heterogeneous one, in all cases, we measure the volume fraction of void, of celestine, and of barite. So in the initial porosity was 86% for the homogeneous, so here 86% at T0 for the homogeneous porous medium, and 87% for the heterogeneous one. And after reaction, the porosity increases to about 90% for the homogeneous porous medium due to dissolution of celestine mostly, and for the heterogeneous porous medium, the porosity decreases to 84%. So clearly here also we can see that we have a greater conversion of celestine to barite in the second case, in the heterogeneous case. And the porosity decrease here after reaction is attributed to the larger molar volume of barite compared to celestine. So finally, we wanted to understand why we have this difference in reactivity, and we attributed this difference in terms of of the flow inside the chip. So to show that, we performed some numerical simulation to assess the local velocities in the microfluidic cells. because even if the fluids and the crystal property and the injection rate is the same, the spatial distribution of the crystals have some consequences, as we will see, on the local flow rate, and then some consequence on the reactivity. So for this simulation, we used the micro-continuum Darcy-Brickman-Stokes approach, where we solve the Stokes equation in the void space, meaning in the fractures, here in light gray. We solve the Darcy's law in the matrix, where we have grains, so here and here in the whole media. And we have the grains, the solid grains that are in white. So here what you see are the images that we use for the numerical simulation. So with this, and we inject the same, the injection rate again was the same, and we just looked here at the velocity field when we do an injection in such media. So what we saw is that for the homogeneous porous media, the mean velocity in the porous matrix is about 9×10⁻⁴ meters per second. And in the heterogeneous media, the velocity is heterogeneous. And we have a fastest flow path in the fractures, along the fractures, with a mean velocity in the fracture that is about 3×10⁻⁵. And in this case, we have a mean velocity in the porous domain that is about 4×10⁵ meters per second. So meaning that with the same injection rate, the mean flow velocity through the crystals, through the porous matrix, is order of magnitude different, depending on whether you have an homogeneous or an heterogeneous distribution of the grains. What we have calculated is actually in the porous domain, we have a factor of about 20 for the velocity between the homogeneous and heterogeneous porous media. And what you have also shown is that in the homogeneous case, the transport is dominated by advection in the crystals, whereas here is dominated by diffusion. So from that, we had those observed higher volume of barite that has precipitated in this case can be explained by the lower velocities, and thus meaning that we have a higher residence time in this case. So to conclude on this part, so with this new kind of microfluidic reactor, and especially thanks to the 3D Raman imaging, we were able to assess the change of mineralogy and structure of the porous media with pretty high spatial and temporal resolution, and to capture local effects, local processes, and to interpret the couplings between chemicals and transport processes. And this setup also allows us to investigate reactive transport in fractured porous media. So with that, this is my final conclusion. I hope that I showed you that experiments in micromodels, when they are associated with high resolution post-processing techniques and with numerical simulation, they allow for a better understanding of the mechanisms at stake. They also allow to provide data to verify and to complete numerical models. And they also allow to improve large-scale models. So with that, I want to thank all the people who have participated in this work, and I want to thank you for your attention. And if you have any question, I will be happy to answer.
Thank you very much, Sophie.
Thank you.
Any questions? Can we see something with the hand?
Björn, do you have any question?
Yes, I would very much like to have another question. For the reactive flow experiments,
Yes.
I don't know exactly what the reaction mechanisms are, but I guess you have to understand very well how the reaction actually takes place, because when you inject barium chloride solution into a saturated strontium sulfate solution, I guess it's the fluid phase is then saturated with sulfate ions that would react with the injected barium ions. So the reaction doesn't take place between the solid strontium sulfate and the barium chloride, but rather the sulfate ions in solution.
So actually, maybe I forgot to mention it, but before injecting the barium chloride, we injected water before starting the experiment to be sure that, so we injected water, pure water, to flush the solution of strontium sulfate and to start with. So yeah, maybe I forgot to mention it.
Okay, I didn't catch that. Now, what about if you do it in the opposite way? In other words, you have a suspension of barium sulfate particles or crystals in
Yes.
the reactor, and then inject a strontium chloride solution. I guess that would make a difference, and I suppose the solvation product of barium, or the solubility product of barium sulfate, is much smaller than that of strontium sulfate. I don't know, but I guess
so. Yeah, in this case, you will expect to have a decrease in the porosity if you replace all the barium by strontium.
Yeah.
Okay.
Okay, thank you anyway. Very interesting talk.
Thank you.
Okay, thank you. Thank you, Sophie. If there's no more questions, then we will end up the session today. Okay, thank you.
Thank you.
Bye.
Bye.